Hey there, science explorers. Today we're embarking on a fun little journey through the fascinating worlds of biology, chemistry, and physics, all bundled into one lighthearted adventure. First off, let's talk about stress. Not the kind that makes you want to hide under your bed, but the biological kind that your body experiences when things get a bit hectic. Stress can actually influence your metabolic pathways, those intricate routes your body uses to turn food into energy and building blocks for life. Imagine your body as a bustling city with each metabolic pathway like a busy highway, delivering essential molecules to keep everything running smoothly. Speaking of molecules, let's meet some tiny characters. Phenotypes: histamine, hippurate, tyrosine, fumarate, thymidine, allantoin, galactose, glucose, hydroxyacetone, guanidinoacetate, glycine, dimethylene, acetylcysteine, and theanine. Sounds like a mouthful. These are all molecules involved in various biochemical processes. For instance, glucose is the body's favorite fuel, while histamine plays a role in immune responses. It's your body's tiny alarm bell. Tyrosine and fumarate are part of the pathways that help produce neurotransmitters and energy. Speaking of neurotransmitters, let's dive into the nervous system. Imagine neurons as busy messengers transmitting info via electrical signals. These neurons have dendrites to receive signals, a soma as their control center, and axons that act like highways guiding messages to terminals where communication happens at synapses. When a neuron gets excited, an action potential, a tiny electrical pulse, travels down the axon, causing neurotransmitters like gamma-aminobutyric acid, glutamate, and dopamine to be released. These chemical messengers then jump across synapses, carrying signals to other neurons or target tissues. How do these electrical signals work? Think of neurons as electrical circuits with membrane potentials acting like voltage levels that fluctuate as ions move through channels. When a neuron depolarizes, meaning its inside becomes less negative, it can fire an action potential, like flipping a switch. And just like a musical instrument responds to light, nerve cells can be influenced by light-sensitive proteins called ion channels. Enter optogenetics, the coolest toolkit in neuroscience. It uses light to control neurons with precision. Imagine shining a flashlight on a neuron, causing light-gated ion channels like channel rhodopsin 2 to open. These are special proteins that act as tiny gates, letting in ions when illuminated, which then depolarizes the neuron and triggers an action potential. This is like turning neurons on and off with a remote control. No surgery required. So next time you see a neuron firing or a glowing fluorescence in a lab, remember, behind that tiny spark lies the universe of wonder. Maybe someday we'll be controlling our brains with just a flick of a switch or a flash of a light.